So there are several modeling methods available that we can use to model biological systems. So at the highest level of modeling methods is statistical mining. So this level of modeling, we can only have qualitative information. So we need, when you want to consider protein localization such as in the membrane and cytosol, we need to include more detailed modeling methods that can give information in terms of structure and spatial localization. For such kind of information, there are several available modeling methods. This includes partial differential equations, spatial GSP, lattice-based, Brownian dynamics. Of course, we can also use molecular dynamics, but molecular dynamics is not suitable because it requires a lot of computational time. So when you want to simulate a model such as MINDI system, it takes about 20 minutes. Molecular dynamics becomes intractable. So we need to use a bit more coarse-grained simulation method. So in our MINDI system, we know that the molecules are involved in both cytosol and also on the membrane. So it has to be able to model both surface and volume reaction diffusion processes. It also has to be stochastic because the number of molecules involved in the MINDI system are very small, only about 800 and 1,000 for MINDI and MINDI respectively. And it also has to include molecular crowding because in vivo, as we know, there are a lot of molecules, a lot of macromolecules that produce a crowded condition in the cell. So this molecular crowding can also brought into effect excluded volume, which can change the reaction kinetics. The modeling method should also be fast. If possible, it should be event-driven, so that only when the computation is necessary, we perform the reactions or diffusion. And if possible, it should be able to include polymerization, because in the MINDI system, as I showed just now, the FTSZ protein polymerizes, and MINDI protein also polymerizes. So in order to accurately reproduce the in vivo dynamics, we need to be able to have such kind of requirements. So what are the available modeling methods, spatial modeling methods at present? So there are many available methods, but so far in this work, my work, I was able to reproduce. I was able to create a model that can fulfill some of the main requirements, such as stochasticity, excluded volume, and surface volume reaction diffusion. So in order to reduce the computational time, I discretized this place into hexagonal close-packed lattice. So in this lattice of equal sized sphere voxels, there are 74%. The 74% of the volume is packed by the voxels, and each voxel has 12 adjoining neighbors. So this voxel here, you can see that it has 12 neighbors. So the reason why I use this hexagonal close-packed lattice is because it is the highest packed lattice available. So this is important so that there's more fine-grainedness in terms of the movement of the molecule. And then it's also useful to use a lattice-based method because we can reduce the computational time by using lipid molecules to represent different compartments, to delineate different compartments. So here you can see the teal voxels are occupied by lipid molecules to represent the cell membrane, and then the maroon voxels are also occupied by lipid molecules to represent the nuclear membrane. So by this delineation, we don't need to perform a lot of computation in terms of to determine the geometry of the cell. So how does, in this method, how does the molecule diffuse? Here just the basis. Molecule walks randomly, a molecule in the cell or in a solution or on the membrane walks randomly, and the displacement is the actual distance between the origin and the time and the end position of the molecule in a given interval tau. So from the Einstein-Small-Minkowski's expression for 3D diffusion, the mean squared displacement is given by 6d tau. So using this equation, so the mean squared displacement is given by the 2 times the radius, which is the diameter of the voxel. We can obtain the relationship between the tau and the movement for a molecule from one voxel to another. So this tau is the time step for a molecule to move from one voxel to another. So now, for the reactions, according to the Collins and Kimball theory, each time a pair of reactants collide, there is a finite probability P for the reaction to occur. So we need to derive the relationship between P and K, which is the here should be the intrinsic reaction rate constant in the H3 lattice. Because as we know the intrinsic reaction rate, but we wouldn't know we need to make sure the probability and intrinsic reaction rate has a valid relationship. So I derived the relationship between P and the K for different kind of reactions. For example, volume-volume reaction, for different kind of molecules, for homodimerization, for surface-surface reactions, and for surface reaction where it involves homodimerization, for intercompartmental reactions such as volume-surface reactions. So next, I wanted to make sure that the method is correct. So first I validated the diffusion in both volume and surface compartments. So here you can see that the diffusion agrees correctly with the predicted, predicted values, analytical values. So this axis shows the time, and here shows the mean square displacement of a molecule. So out of 1,000, when I made the iterations for 10,000, they agree very nicely with the analytical solution. So here the radius of the voxels that I used was 3 nanometer, and surface area was 6 micrometer square, and the volume was about 1 micrometer cube. So I used the periodic boundary condition in order to validate this diffusion. So next, I wanted to validate irreversible reaction that takes place in volume compartment. So this is the irreversible reaction, A plus B to C. So here you can, to compare the simulation, here I compared with the prediction. Prediction is based on the MPK1 theory. So on the y-axis shows the survival probability of molecule A. Survival probability is whether the molecule exists at a given time, and the x-axis shows the time. So as you can see for different diffusion coefficients and different p-values, for irreversible reaction in volume, my method, the spatial side method, agrees nicely. Similarly, for surface irreversible reaction, I also obtained similar results. And next, I also